Seems like the PowerPoint doesn't work again. So let's use PDF. All right, let's get started. So, actually, last lecture we talked a lot about the biotechnology and what kind of genomics data we're getting from, especially the techniques that used to probe the state of our genome, whether there's protein binding, whether it's open or not, or whether there are certain chromatin mark or any kind of histone binding to the DNA. Just want to ask a quick question, please raise your hand if you think I'm going too fast or too slow. And if you want more biology or more machine learning. Okay, cool. So yeah, we're going to talk about more machine learning this time for sure. But I think before going to that, there's one last important technology that I didn't get time to introduce, which is RNA-seq. So basically, with this DNA probing technologies, we're able to probe the state of the DNA at different regulatory process. But what's important is also to know what our cell is expressing, right? So one way of knowing what the cell is expressing is to monitor what RNA you have, because mRNA are the ones that being transcribed and then eventually translated to protein. So I think that there's a very smart technology originating from this mindset where we can, instead of measuring the DNA, we can measuring the RNA in our body, and therefore we can get a profile of, for each cell, what they're expressing at different time and different conditions. So I think there are a lot of details, but I think in short, we can imagine as long as we can sequence DNA, we can always make RNA into DNA by doing what we call reverse transcription, PCR. So we kind of base pairing with RNA, and then you can get a DNA that has the complementary sequence information. And once we get a DNA, we'll be able to use all the technique we introduced last time to get the profile or the read counts of these RNAs. But I think one interesting technique that kind of sparked the huge amount of machine learning in this area is called single cell, because when we're studying expression, actually, if you just take a bulk of cells from our body, they're never going to be exactly the same. They might be in different metabolite state. They even might come from different types. So it's really, if we mix them up together, you will get a very noisy signal. In this example, if each of them has their unique profile like this, but if you measure all of them as a bulk together and then do the histogram, you will get the average signal of all of the four types of cells, which basically you lose a lot of information about what each of them is doing at the moment. So single cell RNA-seq technique is the advanced technology that will enable us to measure these read counts in per cell resolution. So we don't lose track of what cell is expressing what. And it is very genius. So basically, in a high level, it will engineer a lot of these gel beads where each of them has a pre-installed barcode, and these barcodes are unique and they're kind of high throughput enough, so you can use them to differentiate each single cell that you're going to measure. So before we do the sequencing, we kind of pass this bead and the cells through some equipment, and then each single cell will be captured by one of these beads, and they will have this barcode attached to the particular cell, right? And so when we do these reverse transcription, and when all of these reads we get, it has this barcode inserted already. So when we do the sequencing, even though we mix them up together, we can still tell which ones from what cell by looking at their barcode. So this is kind of a high-level concept of what single cell is doing. And actually, it's very hard to engineer this single cell readout system, and it takes many years. Actually, not that many, but it takes years for different technology and different innovation to happen to enable us to do the kind of throughput we are in today. Specifically, I think this is 10x genomics, which is the one of the commonly used technique you will see in the single-cell RNA-seq experiments. So with this developed plate, which each of them can, or we call it chip, each of them can handle 200,000 cells, and we can sequence them all together and get readout from 200,000 cells, and then the total sequencing time is usually just 1 day. So it's really an amazing fast turnaround time. So what we need is to kind of construct the pool of cells that we want, and we kind of treat the cells with whatever condition we want, and then we just put them on the single cell chip, and we get a readout for a lot of them together. So wow, this is amazing, right? So at the end of the experiment, we will be able to measure very high dimensional readouts of basically kind of 20,000 of genes that we have. We can measure the expression of each one of them in each single one of the cells under different conditions. So we'll be expecting to get this kind of data from this kind of experiment where you have this huge matrix where the X or Y dimension will be the number of experiment or conditions you treat different cell. It can be also different cell types, or it can also be different location in the cell or different cell types that you sequence, as long as you can keep track of them. And then on the other axis, you will have the expression level, either normalized or just unnormalized read counts for different genes that we keep track of. So you can see that this matrix is huge, and it can contain a lot of noise. It can also contain a lot of background information. You can get a strong batch effect just by doing the same experiment twice at different locations. You might get some noise and different data. So there's a lot of machine learning or kind of data engineering technique that goes into processing and denoising this kind of data and try to get a clean signal or statistical power from it. And then we can do kind of supervised learning, where we try to, based on this two-dimensional data, we can try to classify or predict like cell conditions, or try to predict like something related to genotype or disease. Or we can also do unsupervised learning just to figure out what are the structure, like hidden structure and hierarchy in this platform, in this pool of cells. So, so I'm not going to talk a lot about single cell, especially for this class, because we're going to start from functional genomics first. But I think this is an area that is very active, so I think we'll eventually talk a little bit about it, and you're also encouraged to kind of do some independent readings about single cell and how these data can be used. Right, so yeah, this is kind of a visualization of different types of analysis of, for example, single cell sequencing of our brain, part of our brain region. You can see that like our brain is really multifunctional, and each of these specialized neuron has different expression. Similarly for other types of human cells, right? So even if like we're coming from like the same kind, there's still slightly differentiation in how the cells are expressing based on different people's genotype and their backgrounds. So combining all of these techniques together, basically we can consider genomics in this era being the studying of this multimodal data, where we have different kind of one-dimensional sequence of read counts, or kind of discrete but kind of a real value measure. And we also have this sequence, DNA sequence or nucleotide, which we can represent either with one-hot encoding or some other types of embedding. And we also have these single-cell RNA-seq that tells us about the expression of different genes at different time. So how we make use of different data to come up with machine learning solutions is kind of one of the theme in the functional genomics area. Okay, so let's talk about the model that we use to do machine learning on it. So surprisingly, if we look at the history of deep learning for regulatory genomics, you will surprisingly find a lot of the fundamental work are built with these architectures, specifically convolutional neural networks, and in combination with some residual networks. And I will explain, and we'll try to motivate why it happens in this lecture. So in general, like not just in Compile, but in general, like when you go to like computer vision class or deep learning class, this concept of representation learning always pop up at the beginning. So basically, we kind of believe that the power of modern deep learning is mostly coming from this hierarchical, the capability of doing the hierarchical extraction of signal such that we can learn good representations from the data, which is slightly diverging from kind of classical deep neural networks when we consider this kind of fully connected neural networks, which really just try to think about like how we can manage complex nonlinear layers and increasing the capacity of simpler models. So I think this representation learning concept is highly relevant to different domains, not just Compile, but also computer vision or NLP and many other domains. Interestingly, this is taken from a slide from Kai-Ming. One of the very earlier and foundational conference for deep learning is called International Conference on Learning Representation. So you can see how representation shows up and why it's kind of considered as the basics of deep learning. And actually, the conference is accepting papers, and it's due early next month. So if you're interested, you can have a try. But what does representation really mean here, right? It's kind of a vague concept that everyone heard about it, like everyone's talking about it. But I don't think there's a one answer. But I think in general, it kind of means the process of converting or representing the raw complex data to solve problems where when we represented this data, we try to kind of use this hierarchy of abstraction layers. So we go from the very raw input into some concept, complex concept, going through this hierarchy of abstraction layers. So you can think of it as kind of information compression or conceptualization, where you kind of extract concept from the data, or considering like doing some abstraction. And then the raw data comes in very different forms. You probably, if you do like NLP and computer vision, they usually come with this two-dimensional grid or a one-dimensional sequence. But in biology, there are also like similar types of data, but also data that are more flexible, like graphs. And so taking an analogy in computer vision, prior to deep learning, a lot of the computer vision work are already focusing on how we can get a good representation of image. So this is particularly done by kind of handcrafting this very delicate filters or feature engineering that try to find some abstraction from the image data, such as defining what is the edge, what is the histogram of different types of edges, and how we can find structures in these low-level engineered features. So there's I guess a decade of work that's just try to do each of these steps differently without using deep learning. However, when deep learning when convolutional neural net come out, it's just surprisingly powerful, and we kind of realize that instead of doing all of these things by hand, we should just rely on the data and let the let the model to figure out what kind of feature what kind of representation is the best for these tasks that we care about. So, the high level of this representation learning in computer vision is really this capability of directly learning these different levels of features or representations from the raw data without needing any handcrafting pre-processing. So it is very obvious in computer vision. Like if you look at these feature maps in different layers of the neural network, you'll be able to see that with this proper training and backpropagation, we actually see that the model is kind of extracting from the low-level features to the more abstract mid-level features, and eventually more complex high-level features that looks like something we can recognize, like a face. So if we think about the DNA sequence, it's actually very similar, right? So that kind of in this huge chunk of informations, we kind of already know that some of it is in charge of coding the protein that will be functioning, fulfilling their biological functions. We'll also know that there's some smaller unit like motifs that will engage different types of proteins and regulators. And so if we think of these small motifs or sequence patterns, which are basically recurring multiple places in our genome repeatedly, we can think of these as low-level features. And and then on a higher level, during this gene regulatory process, not just a single motif or single transcription factors, but also like a combination of transcription factors and histone marks and also chromatins will engage in this process. So it's not relying on just a single motif, but a combination of motifs where these motifs can be placed in the different distance or positions in the genome. And so therefore we have this kind of hierarchy of features in DNA sequence as well. And it's really hard to handcraft a lot of these. So a lot of the kind of prior to deep learning era, we kind of did a similar thing as computer vision, where we just try to directly engineer these motifs, where we look at the data and we try to figure out what are the common patterns in the sequence directly, and we come up with these motif finders or expectation maximization algorithm. Basically, we kind of try to do a similar thing what the vision people do by engineering these filters, right? So, but with the deep learning, we actually get a huge boost in capability of capturing more complex features by directly learning them from the data. Any questions so far? Cool. So this is kind of one of the motivations. So now we can try to motivate the convolutional neural network architecture more, right? So if we think about the fully connected neural net, which is kind of the origin of all neural networks, although it is shown to be very powerful in terms of having a super high VC dimension, or you can prove like with elevated width or depths, you can get the universal function approximator for like polynomial functions. However, if you look at how this neural network works, right? First, it starts from making your data into this high-dimensional vectors or matrix, and each of them is fed into this neural network as one input to one neuron. So what it does is connecting all neurons in the hidden layers to all neurons in the input layer. So each of the pairs gets a distinct weight, and there's no any notion of ordering or spatial information here, because if you swap x1 x2, you can still make similar prediction if you just swap the weight matrix. Right? It doesn't matter, like these two are the same thing. And then it's just too inefficient, because if you have like n dimension of data and h dimension of hidden neurons, you have n times h number of parameters, and these parameters are just kind of wasted if they're not effectively activating all of them. So I think all of these deficiency of fully connected neural network really inspired this invention of convolutional neural networks, which try to focus on one type of inductive bias. By saying inductive bias previously, I have explained this concept where you have kind of a model family where the function space that your model family is modeling have a particular distribution, right? So what convolutional neural network is having a high density in their function space is basically all functions that is able to focusing on this local computation and focusing on this local spatial structure, which are kind of translation invariant. And the key contribution of convolution, the key advantage of convolutional neural network is this parameter sharing mechanism, where basically it reminds us that you don't have to have all each of these pair of edges to use to have its own specific weight. In fact, if something is positional invariant, you should be able to share the weights when you look at the pattern at different positions in the image. So this parameter sharing really makes the convolutional neural network parameter efficient and very expressive in this kind of data. So more particularly, what the convolution means is basically you have the input. Here I'm still using image as an example, but you can imagine like any kind of 1D, 2D, 3D matrix, as long as you can represent it as a matrix. First we'll need to define a concept of filter, which is the a weight matrix of a size, particular size. In this case is 4×4. So we're using this 4×4 weight matrix, and we apply this weight over a small patch of image of the input, which also have the same shape with this filter. And we can use multiple filters to extract different features if we want. And most importantly, we'll be reusing the same weight for different patches in the image by kind of sliding these filters through the image with some step size, or we call stride. So this patchify operation and kind of patch-specific linear operation is called convolution. And we can call each of these weight matrix a convolutional filter. Any questions so far? Cool. So if we look at how this operation is conducted, it's really just element-wise multiplication and summation. So if we're going to use this filter over this example, what we do is we just multiply this matrix with the first patch position-wise, and then we will be summing up all the positions that are positive. Then we'll get number 4 for this particular filter.
How are the filter weights initialized?
Oh yeah, I'm gonna definitely talk about it. So yeah, I think it's very important, and there are different techniques in deciding how we initialize. But you can imagine like normal distribution being one of such, where you want to do like zero-centered initialization, and in certain technique we also want to control the variance of the initialization. So yeah, I wanted to use the GIF, but I'm not able to use PowerPoint, so I hope everyone gets what convolution means. If anyone's not familiar and want me to do a more detailed demonstration, please raise your hand. And right, so everyone's comfortable with the concept of convolution? Great. So yeah, so this is kind of the basic unit where we, as we mentioned earlier, in the representation learning, we try to start from learning these low-level feature maps. So after the first step of learning the feature maps, you can see that all of the operations here are still linear, so it's not yet expressive enough as the traditional neural networks, but it's very easy to add nonlinear just by adding like nonlinear activation unit after the convolution is done, right? So a typical kind of one-layer convolutional neural network will have the convolutional filter as the first piece, and then the nonlinearity as the second piece, and then it followed by a pooling layer, which is very important for convolutional neural network, which is basically what makes it efficient, but at the same time try to capturing the spatial invariance. And after this max pooling, we'll be able to downsample our activation into smaller sizes, and then we can then use the fully connected layer as the last layer to learn these complex interactions between the final activations. So this is a more detailed breakdown of what the intermediate layer look like after a convolution. So yeah, I think basically you can consider you have a given size of the given size of your image or your input from the previous layer, and you have this h, which is the kind of or the depth dimension, which is the number of channels that you have. So for the first layer, you have RGB 3 channels, but in the later layers, you will have more channels because it depends on the number of filters you use in the previous layers. And there's the concept of stride, which is how basically step size you use when you scan through this image. And the larger stride size you use, you're kind of doing larger jumps and in your image, so you're kind of skipping some of the kind of local information. And the other concept is the receptive field, which basically for each neuron, you can see that only a small set of weight is connected to that neuron. So it's only looking at the particular region in the previous feature map. But if you do multiple layers and you can backtrack like what each of these neurons connected to the layer before it, you'll be able to get the final kind of amplified receptive field for each intermediate neuron in the neural network. And again, like nonlinearity is kind of what make deep neural networks powerful. So in this case, you can consider that as being try to filter out the negative activation information from this map. And we can definitely replace ReLU with other activation depending on what kind of application we're thinking about. So now pooling. So now I'll talk about pooling, right? So pooling is basically considered as a downsampling technique that try to capture the information from the global feature map, but also at the same time try to reduce the amount of information into a smaller size. So it does two things. The first is reduce dimensionality, and the second thing is it needs to keep the spatial invariance. Well, spatial invariance here just means if you have a pattern that appears at different position in your input, it shouldn't change the output after the pooling layer, right? So, of course, there are different types of invariant operators that we can use, but like if on top of the head, two of such operator we can think about is the max operator and min operator, right? Each of these does not rely on what the location where these patterns emerge. So, so in this case, we're kind of taking for each of these pools, we're taking, if doing max pooling, we're taking the max value out of the 4 in this feature map, and we're only keeping that one in the next layer. So we have this effectively reduce of dimension from 4 by 4 to 2 by 2. Any questions so far?
Yeah, I always wondered with that, you're losing so much information. I know you're trying to reduce the dimensionality, but there's always different pooling techniques.
Yes.
So why do they seem to focus on max pooling so much?
Sorry. Yeah, I think.
It seems like you're losing the most information from max pooling.
I think you can see you're losing information, or you can see you're filtering like unimportant information, right? So I should have shown you a genomic example, right? So in our, assuming we're doing like this peak calling on our genome, and we can see that actually a lot of the positions in our gene, there is no signal at all for DNA activities, right? So there are actually the peaks or the positive signals are quite sparse in our genome. So I think this kind of max pooling mechanisms really is able to kind of capture where the positive signals are, given like a long sequence of DNA, and try to focus on these patterns that are important and kind of repetitively appearing. So you don't need to fit your parameters into those unimportant information, which are basically nonsense or does not have any meaning. Right, and I think it's a great question. I think this particular pooling is still not, you don't lose all the information because you can still see that we're partitioning our image. When we do this pooling, we're not pooling everything together, right? So there's a choice that you can make, basically how big of the pooling size you want to pool, which is basically how drastically you want to downsample in your image. And I'm going to show in a minute another way of doing this downsampling with another type of convolutional architectures. But I think in computer vision, you could basically think about redundant information in in neighborhood of pixels, right? So if we do kind of if a high resolution image and low resolution image, the low resolution image is kind of like an average pooling of the high resolution image if you take the small patches in the high resolution one. So overall, the global pattern is still maintained. And I think the good thing about this max pooling is you always keep track of the most important information in each layer until the last layer. Cool, any other questions? Great, so, oh yeah, and I forgot to mention that the max pooling and min pooling are just two ways of doing pooling. There's also other way of pooling where people can pool against the dimension of channels. So instead of doing the spatial pooling, you're kind of doing a channel-wise reduction of the features. And this also happens in some of the interpretable machine learning literatures like RADCAMS. So yeah, I think essentially you kind of basically want to decide where you want to lose that spatial information, because you already have the high-level filters which capture all of the local positional information below it. At some point, you may just decide it doesn't matter where this neuron is. I just want to use the information that there is such pattern in my image. Cool. And so now that we talk about the overall feature extraction layers, then to make it kind of a learning model for a particular task like classification or regression, what we need to do is just plug it into a kind of more conventional neural network, like fully connected layers, or other types of more advanced layers like attentions or transformers, to make the final prediction. So I think the intuition here is basically through these deep layers of convolutions, we will be able to extract the high-level features, which basically the combination between these high-level features are much easier to learn, even with kind of a linear layer. So you will see sometimes people just replace the last layer with a linear layer because they believe that the features are so disentangled in the last layer already, such that you only need to do a linear combination of each of them as the final layer. And this will actually help you organize your model, basically kind of factorizing your model into a linear combination of different important patterns. Yeah, so this is just showing that what kind of task we can do. It's not different from the other machine learning lectures we have in the first class. All right, so there's a great question about whether pooling or this kind of convolution will basically, when you do this convolution, you kind of mix up the information in this patch of image, and you can try to decide at what scale you want to detect the pattern from, right? So if we're doing this compact patch of image where each pixel is exactly connected to each other when we do convolution, we're kind of try to capture really local and small patterns that are kind of focusing on a very small neighborhood of the image. However, there's also research that try to kind of increase our receptive field with the belief that maybe there are redundant information in the consecutive pixels, so we don't have to always just look at the local patterns, but we can kind of enlarge our receptive field a little bit by doing this dilation. So it's actually a very cool technique where if you think about it, you don't increase the capacity, you don't increase the complexity of your model because the number of parameters in each filter is the same, but now these filters are more spread out and they're kind of capturing a little bit more global information as we do it. So you might be asked what happens to this empty, these empty pixels where you're not scanning on them. But actually, if we're doing the convolution with this sliding window behavior, we'll still be able to scan through each one of the signal. We're not kind of skipping any of the pixel. We're just kind of trying to organize the weights. We're try to organize how we spend our weights on when we do when we look at each set of pixels. So, so you can think of this dialytic convolution as doing subsampling of our image first, where the subsampling is happening in a certain ratio and it's happening in a periodical fashion, right? Because when you scan it, you kind of first select this set of pixel and then the next set and then the next set. So you kind of having this kind of sub-selection of pixels depending on the space you put between your dilated convolutional filters. So effectively, you're applying a traditional convolutional filter on the smaller sets of pixels. So basically, for one single pixel, in this case, if you have this stride with a space of 1, sorry, not dilution filter with a space of 1, we will effectively doing convolution on 4 different subsample of the image. And on each 4 of the subsampled image, we're basically applying the normal convolution and then get a normal feature map for this smaller image. And then we can map it back to the full image by doing the reverse interlacing. So yeah, any questions about this? Yeah, I think I have a better demonstration actually. So this is another demonstration using a real image, and you can see, like basically, as you increase the dilation ratio in your convolutional filters, you're effectively doing stronger and stronger subsampling of your image, and you're basically focusing on larger scale information. And what people do is you can actually mix different dilated convolutional filters with different dilation level. So you'll be extracting information at multiple scale and combining all of this multi-scale information together to make a more comprehensive prediction.
Can you show us an example of how CNN is used in the genetic setting?
Yes, I'm going to talk about that after I finish the model architecture part. I hope that's fine. Oh, cool. Right. But just like a brief preview, now that you have this like L by L images, right? We don't really need to fix our input to be this L by L by 3 input matrix. Instead, we can do one-dimensional sequence, which in turn becomes a L by 4 dimensional one-hot matrix. So it's exactly the same thing as if you're doing convolution on the squared matrix, where instead of square matrix, we have like L by 4 matrix, and each position has only one of the element being positive and the other ones being 0. Okay, so now that we know the basic unit of a convolutional neural network, I mean, in the deep learning, we just want to go deeper and magically kind of things builds up and becoming stronger. But how can we build convolutional neural network deeper? It's actually not that trivial because since this nonlinearity goes up, and because we're doing this backpropagation through like a huge number of layers, we're actually facing a lot of difficulties when you train with SGD. And there are a lot of techniques that's being proposed to try to alleviate these issues. For example, by normalizing the input, like if you go to kind of a classical machine learning class, people will say like, if you normalize it by doing like a z-score or min-max normalization, it helps like learn better, right? And then there are two different approach. One is called network initialization, which kind of Anirudh asked about earlier in the class. And there's also normalization modules that drastically help us alleviate the burden when training these deep neural networks. So what happens that kind of stopping us from efficient training? One such difficulty is coming from this phenomenon called vanishing or exploding gradients, right? So this is kind of really simple thought experiment, right? Considering one particular linear layer, if you're only considering all the linear layers in the neural network, right? And we've assumed that x, y, z, where the input, output, and the weight are independent variables, and you can easily compute the variance you get from this output variable y, which is just the variable which is related to the size of your layers, input layers, the variance of your weights, and the variance of input data, right? The variance of input data is something you can't control besides doing the normalization. So after normalization, the variance is fixed, but the variance of a weight can be controlled, and that's where the kind of initialization helps. And once we stack these layers, put aside the nonlinearity for a moment, because the nonlinearity sometimes you can, or like dropout, you can usually effectively consider them as kind of multiplying some constant in front of these variance analysis. We will be having this multiplication effect of variance. Now, because we're doing another layer of y2 equals to w times y, right? So when we stack this layer together, we have this variance of different layers, weights, also multiplied together. So at the end of the deep neural network, the variance of your output is really depending on the multiplication of the variance of the intermediate weight matrices. And similarly, this is the forward formula. We can also do the back propagation to compute the variance of the weight, the gradients, right? So essentially just very similar, but just kind of adding this additional gradients where it's the arrow that you get is the loss function, and its partial derivative with respect to the last layer. So if we look at this variance of the gradients we're getting, right, this is effectively like the dynamic range of the gradients we're getting. Dynamic range meaning like the value of the gradients you're seeing will largely coming from this range or region. So the smaller this is, like the smaller gradients you will get in expectation. And the larger this is, the larger in scale, like you will get in terms of expectation. So if this multiplication of variance is smaller than 1, you will have this kind of vanishing gradient problem because you're multiplying this number smaller than 1 together, which will approaching 0 as you go deeper and deeper. And if you have this variance to be larger than 1, you have kind of exploding gradients phenomenon because you're multiplying something that's bigger. So now we know kind of this variant term really matters. Can we do something about it, right? So this actually motivate the technique of network initialization, where we if we know that this is the good range that we try to target, let's just try to initialize our weights in this range. So at least at the beginning of training, we don't like just randomly go vanishing or exploding. So this is proposed by Javier in 2010. It's actually commonly used in neural network initialization, where we kind of always kind of want to zero center the mean, unless you have particular design or bias you want to track in your model. But we also, in addition to try to control our variance such that the variance times the number of input dimension equals to 1, or the variance with respect to the output dimension is 1. This is assuming that we're using all like linear layers and stacking them together. But there are also like some nonlinear layer like values or dropouts in between. So I think that the math might be a little bit messy, but I think effectively we can consider that all of these value nonlinearity effect effectively act as a reduction of variance by half. So now that you kind of you just need to increasing the variance by 2. So now that you just need to have it such a so multiply this additional constant in the front, and this is called like K-mean initialization. So all of these are kind of implemented in PyTorch already, and usually just when you call like model init, it will like automatically choose these normalization functions. But later in the course, when we talk about like AlphaFold kind of techniques, they're also like sometimes we also want to just directly zero out particular prediction because of the invariance we want to track. In that case, we might also just zero initiate all the weights in part of the neural networks. But that's not related to this inductive bias that try to tackle vanishing or exploding gradients, so I'm not going to talk about it here, right? Cool, so, but is this enough, right? It's actually not enough, because if you initialize your network at the beginning, you only control the variance at the beginning, and you kind of only do it at one time. And it's hard to say what happens during optimization, right? You can still go off track. Is there other ways that can dynamically track these variants in between and get a more regularized gradients, right? So that kind of motivate the other lines of technique called normalization modules, where the key mindset here is we try to normalize throughout the training instead of just at the beginning of the training. And we do it in all layers instead of just the input to the first layers. And then if possible, we kind of want to make everything end-to-end and trainable. So this inspired this kind of normalization module, where the first one is introduced in the ICML in 2015. Basically, it's kind of very straightforward, where you, given an input x, we just try to normalize our input by doing this mean shift and the normalized standardization. So then with each batch of data, we're having a x hat, which is basically more centered and having a controlled variance. However, if we do this, we're kind of losing some degree of freedom. So in the case that we still want to learn about the operations on the original x, we want to add these learnable parameters after the normalization, so we can give back this flexibility in kind of learning this additional transformation of the raw signals. So, so in between these normal layers, like linear layers or nonlinear layers, what we do, we can add these regularization modules, which effectively change our kind of variance without without introducing a decrease in capacity. And depends on how we normalize, but depends on what dimension we take this expectation with respect to. It kind of it kind of will ended up being introducing different types of normalization. For example, the batch normalization. Well, each one of here is kind of a visualization of a batch of intermediate feature map, right? So you can have this data dimension, which is the size of your data. So here is just the height times the width, and you have the channel information, which is how many feature maps you have, especially when using convolutional neural networks, or like how many hidden units you have. And then this n dimension is the number of data. So in the mini batches, you will have like maybe a batch size of 32, then you have 32 data in this dimension. So these are kind of the commonly used normalization modules, especially like batch normalization, where you just doing this mean and average and standard deviation with respect to the data dimension. And you have this layer norm where you're kind of doing this per layer in a kind of a channel dimension. And actually layer norm turns out to be very effective later on in these in the modern models we use, for example, in diffusion and region transformers. Also, of course, you can also control like other types of normalization. This all depending on your inductive bias, right? But basically, if you introduce this kind of thing, you will change how your model is likely to perform. So you really need to use your intuition on when you design these. And it turns out that this kind of normalization module really helps boost the training performance. So training now goes faster, and also the overall convergence is just much better before using them. Okay, but is the problem solved? So with all of these techniques, we kind of think maybe we were able to just stack more layers and get more power out of it. However, there's still a period of time where things don't happen as expected, where people find like if you, after kind of this VGG, AlexNet, once people can try to stack more layers into our deep convolutional neural networks, things that's actually got worse and the optimization becomes difficult again. And it's not because of the overfitting, because they kind of train with a huge amount of data, and they have this kind of validation set to observe that it's not like going down and up again. So everything is trained to the best, but still like once you stack more layers, the performance got worse. And this kind of inspired another extension of convolutional neural network, or basically a new type of architecture called residual net, which very effectively solved this problem and enable us to go even deeper afterwards. So before we kind of introduce the network, we kind of want to do like a thought experiment, right? So if we have this shallow network with fewer number of layers, you could effectively, and you have these deep neural networks, right? Basically, the deep neural networks can be considered as this shallow network with additional layers in between, right? So so basically, if we think about these two networks, the deeper network should always kind of capture what the shallow network could capture, right? And you imagine that with the same type of training data, the shallow network should be only kind of one solution to the deeper version of it, because this is more expressive and should be able to learn the things in between, right? So this is what we think about, but empirically, when we do this test, it seems that the optimization of this deep neural network isn't as easy as we thought. But if we already have something that is good enough with a shallower network, why can't we just make sure that when we're adding neural network to it, we're doing this addition of function on top of what I already have and kind of try to give the network an opportunity to fall back to what we already have with higher chance. So I think this is what kind of the kind of the vanilla motivation for residual network to basically give the network an opportunity to fall back to what it can do with the shallower capacity. So considering this normal deep neural network without residual net, and we can consider this function of h of x being what the function space of our current deep neural network is, right? So so basically, given an input x, the family of function we'll get out of these can be written as a h of x. And what we're searching over is this huge family of possible, like this Hilbert space of all possible neural nets that's captured by h of x. What the residual neural network does is, in addition to the original function layers, we add this additional bypass connectivity such that we directly feed the x in the previous layer to the next layer as an addition to the function in between. So now we can write explicitly hx as a function of x plus some other like complex function f of x. Everyone's with me so far? So instead of feeding the h of x, we're kind of feeding the f of x, which can be think of like we're doing kind of a Taylor expansion of the original function x. And then instead of having the model to learn, we already tell the model that you could have this copy of x and you just need to fit on the residual part of the Taylor expansion, right? So, okay, so now you will be worried like, oh, then if we're doing this, are we losing something? Are we kind of losing capacity? But effectively, it's actually not, because if the original shallower network is optimal, then the model will be able to learn to put more weights to the smaller ones and encouraging almost no change to the additional part of fx. So, but if the identity is not optimal, we can increase what this f of x is learning to compensate for the lack of information. So, so basically, we can think of initializing the kind of this additional part with small or almost zero weight. Just try to let it figure out whether this identity is good or not. So effectively, effectively, you can think of this model being not capturing kind of a new functional space, but why is it helping the optimization of neural network? Well, it actually turns out to be like similar analysis we can do on the weight matrix to studying kind of this vanishing gradient problem, where without this residual block, we'll be, again, like relying on these intermediate weight matrices to, in fact, the variance we get from the gradients. But if we have this residual block explicitly introduced in our function space, we'll kind of have this 1 in front of the gradient. So even if the later part is vanishing, we still have this 1 in the front, which kind of stabilize our gradients. And we know that 1 is the good number to have. And that's what all the normalization module is targeting, right? But I think I found this very interesting interpretation from another angle, besides just vanishing gradients. It's actually think about like putting the right inductive bias into the function space. So it's not exactly what's happening in the residual net, but you can think of two classes of functions, where for the first class of functions, you can increase the capacity of your function class by maybe stacking more layers. But when you increase the layers, you kind of have a distribution that is only partially overlapping with your lower class functional class. So when you do this, when you try to enlarge your functional space by increasing the complexities, you will only be able to get close, assuming this f star is the optimal solutions, you'll be only getting close to this f star when you're approaching like the third level of complexity here, right? And then when you further increase it, a lot of time you're actually getting further away from the f star. So it's not anchoring by introducing this kind of effect, you don't have like a good anchor of a good functional space, where if you just blindly increasing the capacity, you might be driven away, like you might be driving yourself away from the getting a good solution, because you spend a decent amount of kind of a weight space or functional class space in exploring the non-useful regions. Versus there's also a nested function classes where kind of perceptually you kind of anchor your function class with the function class with a smaller capacity, such that when you increase the capacity, you're strictly covering what your previous what the previous function class with smaller capacity captures. And this is kind of an interesting illustration of what the residual network might be doing, because effectively we already say that you can always fall back to F1 if you are not satisfied with your F2 with these residual blocks, and just assign it a super small weight, it will fall back to F1. So this kind of, with this anchored nested function class, when you try to increase the function class, you kind of search around what you know that might already be good, and with with the expanding capacity, you're kind of monotonically approaching the good solution without losing the efficiency, without kind of assigning weights to other parts of your model. Any questions with this kind of interpretations? So yeah, so effectively we're not, so considering each of these blocks being kind of a high density region of your model class, it's not just like the only sets of model that's possible in this set, but you put a high weight on this set, right? So when we talk about Bayesian neural network, Bayesian learning long time ago, what we're doing by designing this neural network is we're designing different priors over the architectures. And with the different setting of neural networks, you may be covering same function space, but with a different probability. Right? This is basically capturing what belief you have in this function space. So having this residual block introduce, you kind of increasing the opportunity or probability that you will fall under this nested function class regime, where when you add more layers to it, the expanded class have a high probability of falling exactly on top of the functional class with the smaller capacity. Cool. Okay, so yeah, so now let's now we have introduced these two architectures. We can see why and how this can be applied to functional genomics data. Right, the first simple example is from this kind of the task of motif discovery, right? So we talk about the motif repeatedly before, which is basically the sequence specificity which decides whether or not it will be binding or interacting with certain protein. So for taking the transcription factor as an example, we know that these transcription factors are DNA specific proteins that only interact with certain type of sequence because of the biomolecular conformation of the DNA sequence. So conventionally, we invented this motif, and one way of representing motif is called PSSM. So basically, we compute the weight, the frequency of each nucleotide appearing at each position, assuming kind of independency between different positions, and we also compute the probability of observing such nucleotide in the background distribution, like basically in the average, if you look at the whole genome, what would be the background distribution is, and we take the log of the ratio of the two probability to get kind of a center or normalized probability that tells us how different this distribution is from the background. So, and that gives us the log of the ratio, or log odds gives us these parameters of PSSM. So if you're summing up the letters and its corresponding PSSM factors, you're effectively getting the log of the joint distribution over that particular sequence. And you can measure the probability of that sequence coming from this motif versus the probability that this is an average sequence. So what we're effectively doing is by taking, for example, if you have the sequence GCATT, and we want to compute the PSM score for this sequence, we just look for the weights for that particular sequence, for that nucleotide at that position, and we take out the weight for each of these single nucleotide, and we can just sum them up together, or take the average of them to get the final PSSM score, which effectively tells us the likelihood or log likelihood of this sequence under this particular positional matrix or distribution that we assume for this pattern, right? So we can do this for different positions, which is what we kind of assume that there's invariant motif appearing recursively in the genome, which we already know it's kind of true, because the biophysics between sequence and protein is also kind of fixed, right? So when we do this what we call motif scanning, by scanning these weight matrix across the genome, we're effectively getting these readouts of PSSM score at different positions of the genome. And what we usually do is to do a threshold apply threshold on these scores. So we consider this position as significant, being like basically the probability of motif appearing in this position is much higher than other positions. So when we look at these operations, right, it's basically multiplying particular weight matrix by a one hot matrix where only the position with value 1 have a non-zero value, and the final score is the average of the element-wise multiplication of the PSSM and the one hot short sequence. So this is exactly what convolutional neural network is kind of doing. Just the only difference is instead of doing convolution with a continuous value of RGB values, we're here doing a convolution with either 0 or 1, like 0, 1 matrix. Any questions so far? You can see the kind of the coincidence here where what we're doing with motif scaffolding is kind of exactly what the convolutional filter is computing the feature map from the input sequence. And if we're considering these patterns, like if we're getting these low-level patterns in this feature map, we can further try to learn higher level grammar about the transcription gene regulation by looking at the global pattern of where these small motifs locating. So this is a very common architecture in deep neural networks over genomic sequence, where you're representing the sequence with one hot, and you're applying these filters, which effectively does kind of motif scanning, and you have this motif scanning outcome as the intermediate layers or feature maps. And then on top of that, you'll be able to learn global patterns, which is how these motifs are combined over the entire sequence with this higher level convolutional filters. And then you'll be able to tell, like, this sequence is spaced maybe k number of nucleotide away from the next motif. And with this particular spacing, it is predictive of some final outcome of gene regulation, such as, in this case, it's a transcription factor binding of these two transcription factors. Any questions so far? So the output can be, yeah, actually I'm going to introduce that exactly right now. So the output can be depending on what kind of task we care about, right? So in this case, in this example, is whether or not something is binding to this sequence, whether or not there's a GATA1 or tau transcription factor binding to this region of the genome, which is, I think, maybe 32 a base pair length sequence.
So the input has a label?
Yeah, so I'm going to talk about it right now, right? So this is one way of motivating why using convolutional neural networks for DNAs, but genomic data is not just DNA, and it can be more diverse and actually have multiple modalities, just like image and text. So so this is what we get from all kinds, these different types of probing technologies that we talked about in the last course, right? So eventually, a lot of these technique what you get at the end is on the one-dimensional scale of basic different positions in our genome. We have the sequence that is associated with each position on the x-axis, And then we have the signal or read counts that we observed at each position in the genome, right, which we call it profiles or signals. So you can see that these are kind of continuous value, well, not continuous, but like discrete, but real value numbers. And you can think of it as like a one-dimensional sequence of signals. So usually in the conventional bioinformatics, we would call this technique called peak calling, where when we look at these profiles of activations at different, like read counts at different positions, we would decide whether or not this is actually a real signal and there is something happening here related to the function that you're testing. Maybe it's a DNA-seq, then it means this part there is no chromatin binding. If it's ChIP-seq, it means on this part of the genome there's a protein binding, either a transcription factor or histone. So then we can consider, like, for each position in this genome, we will have two types of signals. One is the continuous value signal of how strong the activity we observe. The other is a binary label, where basically using this peak calling, we'll be able to say, well, there is an event or there isn't an event. So a simple task we can think of is, for sure, like for a given sequence, whether or not there's anything happen in this sequence, right? So that means we can, for any region in the genome, we can take out a sequence with a fixed window size, maybe 100 base pairs, 200 base pair, depends on, or 1,000 base pair, depending on like how big of the neighborhood we're looking at, and we can assign a label to this 100 base pair sequence by saying whether or not there is a peak within that region. Or alternatively, we can also predict what is the average what is the maximum activity you get in this region, right? Or what is the overall like mean recounts that you get from this region. So that will basically becoming a kind of regression task where we're predicting a number. And instead of predicting one value for the entire sequence, the other thing we can do is we can predict per position-wise what the signal is. So instead of recovering the overall like existence of a peak or not, we're kind of predicting at the resolution, at the residual resolution level, the exact signal at each position. So we get output of L by 1 if it's a binary, right? It's a 1, 0, 1, 0, 0, basically annotating the entire sequence. And this can be definitely done with just changing the architecture and the output size of our networks. But besides this DNA sequence, there are actually a lot more information we can use as features, right? Because when we recall what we have what we get out of these genomic data, is not just the sequence, but these 1D vector of features. Why not also use them as the features, right? So we can organically combining the signal information together with the DNA sequence. Both of them are kind of vector, despite that one of them is discrete, the other is more like continuous value. So we can have this multimodal input feature and pass it through similar architectures. And then as an outcome, we can do multiple prediction at the same time, right? We can predict each of them, like doing different combinations. It's all flexible, depending on the problem we're studying. And besides these 1D features, there are actually more complex features and high-resolution features as well. For example, this is kind of a tag-seq data, where we just try to cut the genome where there's nothing binding to it, or there's like no chromatin, kind of try to compress the genome. So once you cut the DNA with a certain type of enzyme, you will have this short fragments that tells you like what kind of protein is binding to that region, where the shorter sequence you have are basically just kind of a loose DNA, open DNA regions, and where if you have this longer region, basically you can imagine that this region is intact because it's wrapped around certain DNA, certain protein like a nucleosome, right? So when we have these kind of plot, we can first plot them into the one-dimensional signal space that will give you this information. The other way we can do is we can also cluster them by their sequence length. So we will also get the frequency of the sequence length from the data. And so interestingly, we can plot both of them in two dimensions. So if we plot the position of the sequence on the x dimension and the length, fragment length distribution on y dimension, we get this two-dimensional like image. It's basically kind of like an image, right? So it has this kind of interesting spatial pattern where kind of in the center of the binding site, you tend to have a shorter fragments because you know that these are the short open DNA region where you are interacting. And then further away, you might have longer sequence. But in between, there's a gap because there's protein binding to it, and you cannot have sequence that are not intact. And then in the further downstream and upstream, there are regions where the whole genome is closed, where your DNA is wrapped around the nucleosome, so the fragment lengths get larger. So the 2D information provides also like a lot of information. So actually, there's a lot of different architectures that you will see through like literature reading on these different tasks. But what they're doing is basically, like I said, a combination of input features and different combination of output features. And CNN is commonly used as the architectures to fulfill this kind of predictions. So one such very famous architecture is called DeepBind. It's actually the first deep learning or convolutional neural network model built for transcription factor binding. It actually came out just in my first year PhD. So that's also like the first paper I wrote about is also about this kind of doing the architecture search as an extension to DeepMind. So like, yeah, I think that the idea is quite simple and it actually worked so well compared to like the conditional classification method like SVM, a K-mer-based approach. So it's kind of opened up this whole field of deep learning for transcription factor binding. And what it does is basically taking the protein binding microarray, which is the experimental data you get, and then you can cut your sequence into a fixed length segments, maybe 200 base pairs. And like I said, in this particular paper, they just kind of assign a single label to each 200 base pair DNA sequence and try to make a prediction of that, like just make a binary prediction whether a transcription factor is binding to it or not. And they also do all kinds of typical machine learning thing where you split your data into a training set, a validation set, and do like architecture search and try to figure out using the validation set what's the best architecture, and then you train it with more data and then do a one-time like test. And you got a really high UROC on some of these tasks, which is amazing. The other thing you can do with this prediction, neural network prediction, is you can try to study whether there's mutational effect in the input sequence just by making mutations to your input sequence and observe the change in predicted value in the outcome. So this will give you kind of an in silico mutagenesis of how each change of amino acid type will potentially impact the transcription binding probability. And they also find some correlation between these analysis and like known variants that relate to human disease. And of course, there are a lot of different extension follow-ups